Okay, now asking the basic idea what we get here. After observing these cells, we observe majorly two structures. That is, one is the boundary of the cell. If you say the cell again here, in this cell there's a boundary, a line we draw in the pencil. That is called the cell membrane. So that is the most basic structure found in all living cells. And the other thing, what you observe in this is there is internal area inside the cell that we call the cytoplasm. So any cell there is a boundary called the cell membrane, and inside the cell membrane you have this called the cytoplasm. Those two structures are very important structures and they are very common for the all living cells, right? That's the thing. That means we are learning a new word now. That is, you have to understand if this is the whole cell, what are these internal structures we called? So we call them cell organelles, not organs, right? Remember the word, don't confuse with organs. These are called organelles, right? These organelles we call the substructures, the components that build up the cell. So what we are going to learn is now we are going to learn each and every cell and their organelles, right? Let's see. This is a typical animal cell. So you can see the cell now, it's a typical animal cell. In this animal cell, we are going to learn what are the structures, or what you call the name, you know the name now, organelles we can see inside the cell. So these are the organelles we can see. You have to remember the shape of the organelle as well as the where it can be seen, right? Start from the top part. Now I will do like this. The basic structure I will go first. You can see here that the black color dot you observed inside the cell is magnified up to this level by using an electron microscope. You can see this thing now very clearly. So that black color dot is having substructures like this. See that? It's a very complex structure. So that whole thing we call the nucleus. Inside that nucleus you have substructures we call, even they are called organelles. The nucleus is made of the nucleolus, that's a central dark color region in the nucleus, and the other thing, the nuclear envelope. There's a cover around the nucleus, but that nuclear cover is not permanent, so you can see small cavities in this. You can see small cavities in this area. So from these cavities, the content inside the nucleus, I will tell what is it, that will communicate with the other parts of the cytoplasm. That means you know that the characters of a cell is governed by DNA and also RNA. So RNA and DNA, we call them nucleic acids. They are found highly inside this area, so we call it chromatin. In the future we will learn in genetics lesson what is chromatin. That chromatin contains DNA and RNA, so these things will go outside the to the cytoplasm and communicate and do functions of the other parts of the cell. That means the nucleus is the major structure inside the cell, major organelle that perform the control and activity functions inside the cell. See that. In that, if you zoom further, you can see here, in that envelope, you can see it's occurring like a segment of tubes. See, a segment of tubes. So these tubes we call reticulums. Endoplasmic reticulums, we call them. But in these endoplasmic reticulums, if you further see, there are small structures attached to that. The tube is there. Into this tube, a small structures are attached. What we call these structures, we call them ribosomes. We will learn in the future, even ribosomes are found in the free space in the cytoplasm also. But these ribosomes are attached to the membrane of the endoplasmic reticulum. Therefore, due to these attachments, this endoplasmic reticulum is further detailed by having a name called rough endoplasmic reticulum. Rough endoplasmic. Why we call it rough? Because there are ribosomes attached to that. So you will think, if it is rough, there should be the opposite one, the smooth one. Yes, of course there is. In the other parts of the cytoplasm, see, nothing is attached to the membrane, so these are smooth tubules. These tubes are called smooth endoplasmic reticulums. We call them smooth endoplasmic reticulums. Right? So you have two categories of reticulums here, smooth and rough endoplasmic reticulums. Right? Next one. You already know this thing. Sometimes you heard about the word. This is the most important organelles in the cell, because everything you know that every living or any kind of machine or something that do the work, they need energy. So there's a structure in many of the cells, but there is a alternation here. That means you have to understand one thing later, that all the cells don't have the mitochondria. Some cells, they generate their energy by the cytoplasm also. But this mitochondria is a common part in the animal cells. Advanced animals, we have it. So mitochondria is a structure like this. You can see that it's having two membranes, double membrane structure. In that double membrane structure, it's having the wavy appearance in the inside the subcytoplasm inside this. So this one is called the mitochondria. Right? One is singular, we call mitochondrion. Plural, we call mitochondria. So this structure is performing the generation of energy by cellular respiration. It does cellular respiration, right? Remember that. So there are many in the mitochondria around the nucleus, you can see, right? Okay. We are learning about the functions later. So just I'm going to discuss these parts, right? Okay. Now the next one. It is a special part that is very important for cell division. So you don't have actually any idea about cell division. I will just briefly say that this thing, if the cell aged, if the cell is having the last phase of its life, this cell start to develop a new cell. Actually what is happening, this cell is divided into two cells. If the cell is divided into two cells, so who will do that activity, dividing the materials inside the nucleus into two? That is done by these centrioles. These are called centrioles. These centrioles are able to divide the parts of the nucleus in equal halves into the new two cells. So remember the name, because in the cell division lesson we are learning about the centriole further. So make the platform clearly. Now I told you about the cytoplasm. That's the area where all the cell organelles are embedded. So cytoplasm is a gelatinous material, like a jelly-like medium, not a hard jelly, a soft jelly-like medium. And the other thing we have to understand is this. You know that imagine this cell is a life unit. That means every function should be there. I told you that this nucleus control the functions. This one generate the energy, the mitochondria, and the tubules. These tubes are transporting substances. I have to say that this is a tube-like structure, because always we know that water board, they transport water to each and every house by using tubes. So same thing happens here. But sometimes assume we have to transport the contents in the cell outside of the cell. We have to transport them to the outside of the cell. So that should be done by another organelle. So because it is division of labor, that means their functions are divide and do. Not the same organelle is doing all the functions. Therefore, it's very efficient. Dividing and rule, it is very efficient. Now, what is the structure? You can see it's like a sandwich slices arranged one by one. It's a stack of tubules. Near these tubules you can see small circular spherical structures. We call them vesicles. So these vesicles are produced by these stack of tubules. We call them Golgi complex, or we call it Golgi bodies sometimes, right? Golgi complex we call this. This Golgi complex is performing the major function that collect the contents from the other parts of the nucleus or the cytoplasm, They collect the contents and they start to synthesize them. That means they produce it according to the pattern and they start to release it to the outside of the cell or inside some parts of the cell. What do you mean by that? For example, assume you are making cake. This Golgi is making cake. It's a factory. So we need ingredients like flour, eggs and all these things. So inside the factory you are going to synthesize the cake or produce the cake, and you are packing it and you are releasing it as the package. That package we call the vacuole. Oh, sorry, vesicle. So that vesicles are surrounded by this thing because it's already doing the function. Sometimes these vesicles go outside. Sometimes they will be remaining inside the cell for further activities. Like that, the Golgi complex is very important for collecting, packaging and distribution of substances. Now our idea. Your first competency is you have to label a cell. Sometimes they give a cell diagram and give us the idea to label it. So let's see how we label it. So I have given you a cell like this, a different cell. I'll give you I will give you some time. You can pause the video. Even by pausing the video, try to label these parts accordingly. You can label it nicely, but see that you always not get the same figure here. You are getting a separate figure here, a different figure in the examination paper, because already students are trying to memorize everything and every shape. That's what they do. But if they get a new type of cell with the same type of organelles, what they do, they start to think about it and they confuse themselves and start to skip this question. But remember, 1, 2, 3, 4, 5, 6, 7, 8, 9. If this is 9 labeled parts, it should be 9 marks. Remember that. So try to do it. Pause the video and try to find out the answers. Right, it's time to check the answers that you have labeled here. In this one, I will help you for this because I didn't discuss about this structure. Usually, you know that in animal cells, the samples or food items or any kind of material is stored inside small sacs. We call them vacuoles, the food vacuoles. So these things are not actually permanently present in animal cells. We can only find them permanently inside inside the plant cell as a large vacuole. But animal cells, temporarily we can find these structures because they will be vanished out, or we should be transported outside the cell later. So this is called a temporary vacuole, or we can say a food vacuole. Then this one, we already know this, a stack of small tubules arranged one by one each other, one over the other. So that is we call the Golgi complex. And the other one, this one, you can see carefully check the arrowheads. This is for not for the boundary, for this structure, the whole structure. So that whole structure we can memorize or recall as the nucleus. Inside the nucleus you have a substructure that is called the nucleolus. And here, so wait, the mitochondria present. This mitochondria is the powerhouse of the cell. And this one, the boundary of the cell, we can see that is the cell membrane, or we call it a plasma membrane. And here also the vacuole, that's why I just didn't ask you what it is. And this one, the tiniest structures, the smallest structures we found in the cell, it's called the ribosome. And this one is the rough endoplasmic reticulum, because how we say it, because there are small granule-like structures we call ribosomes attached to the endoplasmic reticulum, so we call it rough endoplasmic reticulum. And this one, without these tiny ribosomes, this is a smooth one, so this is smooth endoplasmic reticulum. Like that, you are able to identify what are the structures according to the figure they are getting in this diagram. Right, we are shifting to the next part now. Here I mentioned one important thing, that is most of these cells, cell organelles, are not able to observe under the light microscope. You have seen it, only the nucleus which is merely visible, and as well as the boundary you can see with the cytoplasm, that's all. These structures like mitochondria, the Golgi apparatus, the ribosomes, very importantly that vacuole, these structures you can't identify using the light microscope. We have to go to higher magnification, that is done by the electron microscope. So that's the thing. Now this is the plant cell. Even the plant cell has similar structures with the animal cell, but there's a small thing to discuss. That is, you know that plants, if you consider in a basic manner, the plant is able to produce their own food and store the food, as well as the plant are rigid structures. To fulfill these requirements, the plant cell or the plant cells are adapted in different manner. So let's see what are these methods in the discussion now. You can see it now. Other than the structure we found in the animal cell, they are always present in these plant cells. But other than that, you can see a large area is covered by a large sac-like structure we call the central vacuole of the plant, or we call it having two structures in that internal region of that thing we call the cell sap, and the other thing, the boundary which it covers, is called the tonoplast. Altogether we call it the central vacuole. So that is a unique feature found in the plant cell, this one, because I will give the idea that this structure is providing the support, mechanical support, because you know that water is filled in this, water and other materials. So it provides the hydrostatic pressure that we needed to maintain the shape of the cell, as well as other impurities or other kind of storage materials are also absorbed and stored inside this large vacuole. So those are the features of this vacuole. So if you go to the other parts also, mitochondria, Golgi, these things are present. You can see a very thick structure around the cell. There's a very thick and very rigid structure. Even you can see there's a specific shape for the plant cell. So why is that? Because there's a hard material we call cellulose cell wall is present around the cell membrane. So internally, inner region, this thin line is the cell membrane. Outside of the cell membrane, there's another layer that is called the cell wall. The cell wall is made of different materials. I will discuss it later. So I just say the materials now. Cellulose, hemicellulose and pectin, they all together get like you know that one of the concrete mixture, it will start to form a cell wall around this. Don't forget, cell membrane is there. Outside the cell membrane you have the cell wall addition to that. Then and the other part, you know, the plants, I told you the previous also, the plants are able to do photosynthesis. That means they are doing photosynthesis by the help of a special structure, a green color structure found in the cell, plant cell, that is called the chloroplast. Inside the chloroplast, several other structures are there, which is called the chlorophyll pigment, as well as there's a small matrix inside. That matrix is a liquid medium containing large amount of enzymes needed for the activity of the photosynthesis in chloroplast, right? So those two structures, three structures, the vacuole, the chloroplast and the cell wall, are unique characteristics for the plant cell only. You can't find these large vacuole, and again I am telling, large vacuole, chloroplast and the cell wall inside the animal cell. You can't find them. Those are the differences between the plant cell and animal cell. So I have mentioned here the differences clearly. So cell wall is one of the thing, vacuole and the chloroplast. Vacuole, the content is pushed to the corners, to the peripheral regions of the cell, because there's a large central vacuole which is dominating in the center of the cell. Now it's again your chance to label this structure. So I'll give a few minutes, or you can pause the video and start to label this thing for your common knowledge. Right, it's time to discuss this thing. So I hope you have labeled nicely. See this thing. The upper block, you can see it is labeling a one part, that means a solid layer around the cell. That is the cell wall. So this is the cell wall. And the second block, internal to the cell wall, there's a small layer. We already know all the cells are having this structure, that is the cell membrane, or we call plasma membrane. Then this one, see this. You can see a small stack-like structures connected by filaments, and it is already always present in a larger amount in plant cells, that is the chloroplast. don't say chlorophyll, right? So chlorophyll is the green color pigment, but chloroplast is the organelle having the vacuole. So this is the Golgi complex, or we call Golgi apparatus. Next, this one, see the large one. So because the other structures I have already labeled, because you know these things now, so I just want to highlight some of the structures in the plant cell. So this one is the vacuole. So you can see now this is different from the others default typical cell you observed. That's what you get in the exam also. You are not getting the same cell, you are getting different diagram to label. So students should adapt to the question. They will give an argument that in that cell the vacuole was in the center, nucleus was in the corner, so why this is like this. These things are called adaptations and the evolution, and the other thing, the growth of the cell. You can see the large vacuole is subdivided into two. You can see two sections, but it's a large part, right? So that's the vacuole. You can say it's a vacuole. Sometimes this some students will think this the content inside. So cell sap is also applicable or appreciable answer, right? Remember that. And the other thing, see this one. Always remember something is connected to the cell nuclear envelope should have a certain arrangement of ribosomes in this. If you can see, they have arrangement of ribosomes, but you can see that in here. But you can't see ribosome arrangement in this. Therefore, this is a smooth one. So its rough endoplasmic reticulum is here. So this one contains no ribosome, so it is the smooth endoplasmic reticulum. So endoplasmic reticulum is a long word. So normally in biology we say that rough ER. So we can write smooth capital E capital R, ER, right? Next one, this is the nucleolus. This whole thing is the nucleus. Inside the nucleus you have a substructure called the nucleolus. And this is the nucleus, or sometimes some students confuse that as the envelope. So I shall give marks for that also. This will be the nuclear envelope. So it's a nucleus or nuclear envelope, right? Okay. So hope you got all correct in here. So that's what we call the progress and satisfaction, right? Okay, let's see the next one. You can see this table. I summarize all the functions of each and every structure in the plant and animal cell. So you are going to find out what are the organelles given in here. Before that, before we do this activity, you are going to see these things about different structures in the plant and animal cell and their function. So 50% of the knowledge is there. You know the structure now. Only thing is you have to discuss about the function. Start from the cell membrane. Cell membrane is a structure which is enclosing the cell. Always all the cells they have the cell membrane. And the other thing, allow changes of exchange of water and other substances actually. So it's called selectively permeable or partially permeable, because the reason it will not allow everything to come in and go out of the cell, because it should control, otherwise the cell is not having proper life inside the cell. So it should maintain the life inside the cell, so it has a control of entry and exit of substances. So we call it selectively permeable or partially permeable membrane. Permeable means it gives the permission to go. So it is partially permeable, that means it will not give the chance for everything to go in and come in and go out of the cell. And we have to find out what are the two things which is made of. It's made of not only two things, there are many substances, but basically the cell membrane is a fluid-like structure, semi-solid structure, which is like you know that, like jelly-like structure. So that structure is made of two materials, mainly two materials, that is phospholipid and proteins. They get together and make it by the help of other materials also, which is not discussed in this syllabus, right? Because you have to have the frame inside the limit. So in that limit you are learning only two materials, main materials, that's called phospholipids and proteins. They build up the cell membrane. Next, you can see the structure provide mechanical support and protection to the cell. That means the kind of a structure which is providing support, but we have to understand what is the What are the features of this? Outer covering of plant cells. It is only found in plant cells. That means it made of cellulose, hemicellulose and pectin, relatively rigid structure. So this will be the cell wall, right? It's a cell wall. So next one, we can go to the next one. But before we go to that, if we go to the cell wall, we have to think one thing, that that cell membrane control the entry and exit of substances, allow the exchange of water. Even cell wall is passing water inside and outside. It's not having any obstacle to pass the cell wall. Otherwise you will think that a wall, like a brick wall, you can't pass through anything from that. But it's not like that. Water and some minerals can go through the cell wall, but not through the cell membrane. Cell membrane will allow it to go when it is required, not just excessively go through it. That means you can finally understand that cell wall is fully permeable for substances. Right. Next one. Cell oval rod-shaped structure. Double membrane bounded organelle. Oval shape or rod shaped structure. Oval rod shaped structure and double membrane organelle. And from that you can't find out what is the structure. But if you go to the function, production of energy, if they say this thing, ultimately quickly you are getting the answer, that is the mitochondria. In that they are saying the metabolic activities. What do you mean by that? Metabolic activities means biochemical reactions that happens inside the living cell. So for these metabolic activities, the mitochondria is providing energy by respiration. You will learn in the future what are these respiration and what do you mean by that thing. Then the next one, flat tubular network of sacs contain attached ribosomes in the membrane. Contain attached ribosomes in the membrane. Then you can understand. From that sentence you can catch what is the structure, and also for further details, it should have tubular shape. So it's the endoplasmic reticulum. But what is it? What the adjective we put? It's the rough endoplasmic reticulum. So when we go to the function of this, it is containing ribosomes. The major function of ribosomes is to synthesize proteins. Ribosomes take the amino acids. It's a building block of proteins. They take all the amino acids and make them synthesize a large chain of protein, a polypeptide. So that is done by ribosomes, and these ribosomes are attached to the endoplasmic reticulum. So reticulum is able to transport these proteins throughout the cell. So that's the united function between the ribosomes and the endoplasmic reticulum in RER. RER means rough endoplasmic reticulum. Next structure. They are telling here fluid-filled large organelle commonly found in the plant cell. Surrounding membrane is called the chromatin. From that we can get this thing. This will be the central vacuole, the large vacuole inside the plant cell. So these are the things you understand in that. And also inside that vacuole you have cell sap. It's a liquid medium. What it contains? It contains water, sugar, pigments. What are pigments? Pigments are colored substances. You know that how the petals of flowers are getting colors, nice colors, different colors. So these pigments are found in the vacuole inside. Even some other metabolic substances, metabolites, like that when you crush some leaves in some plants, you will get unpleasant or pleasant smell. So these called tannins. So these tannin like hydrocarbon substances are also found inside the vacuole. And the other thing, sometimes found in animal cells. So this will be actually the vacuole. So if we go to the other parts, other structures, when we are labeling this thing, maintain water balance, that's the main function of this vacuole. And the other thing, support and function. The function word is missing here. Support and function word is not appropriate here. You can apply the word storage. Support and storage, right? Remember those things. Next structure, gelatinous liquid medium. Gelatinous liquid medium. From that you can't find out what it is. We go to the second quality, second feature. Organic and inorganic substances are present. If contains organic and inorganic substances, even you can't find out what is the structure. But one thing we can mention thing, that is gelatinous liquid medium. That means it should be a medium for others. That means you get the idea. This is the cytoplasm, right? Maintain the shape of the animal cell, because cytoplasm gives the support to the animal cell. Only that thing, because there's no cell wall for the animal cell. Carry out all metabolic activities in the cell. That means it's a medium where contains water and other things, substances, nutrients, minerals and everything. So what will happen in here, they will get all the contents nicely. Next one, you can see this structure surrounded by a nuclear envelope. Nucleolus and chromatin body is present. See that. Ultimately, you know what it is, the nucleus. Nucleus contains an envelope as well as in the central area. You can find the nucleolus and the chromatin. Let's go to the function, because you have to understand the main function of the greater structure inside the cell. The nucleus contains DNA. In the nucleolus, it contains RNA. Do you know what is DNA and RNA? Because you have learned about these structures in biochemical ideas in first lesson in grade 10. So these are nucleic acids. So chromatin is based on proteinous material attached to DNA. That is chromatin. So it is a network of small threads like structures. They will get contents when it is required for a certain purpose. We will learn that purpose later. Just remember that structure is contained in DNA. That means DNA is a material that stores genetic information of a person or an individual. So when it want to transfer it to the next generation, the nucleus will multiply the number of DNA molecules into twice of it and divide that into 2. Then new cells are formed, and these new cells are getting the equal amount of genetic information, what we call it DNA. And the other thing, RNA. RNA is stored and produced inside the nucleolus. So even today we are talking these things because you know the PCR test. PCR means polymerase chain reaction. What they do there? How we detect that the patient is having coronavirus in the body by using PCR test. We have to understand this thing. PCR, polymerase chain reaction. You take a sample from the patient's body. Actually, you are taking sputum. Sputum means mucus producing in the nostril area or the throat of the patient. A slimy substance, we call it the phlegm. You take a small sample by using a small probe, and you separate the DNA inside that material. If it is there, there should be DNA, because DNA or RNA doesn't matter. Then what they do? They match with their own their original sample of coronavirus with the sample taken from the patient. If both samples contain, if that sample taken from the patient contains the similar RNA inside that means that is containing a viral particle. Now, membrane bound sacs stacked on top each other, and the other thing, associate with secretory vesicles. Membrane bounded sacs stocked on top of each other, so you can think about it. And the other evidence that confirm your idea is associate with secretory vesicles. So this is the structure we call the Golgi complex. Collecting, packaging and distribution secretory substances is the main function of the Golgi apparatus. Collecting, packaging and distributing secretory substances. Secretory means they release the substances outside of the cell. Last one. Organelle without a membrane consists of small and a larger unit. That's a very advanced thing. Found freely or attached to endoplasmic reticulum. So what it is? That is the ribosome. And the main function already know that is the synthesis of proteins. It synthesizes proteins. So like that, I hope that you can now find out the structure relevant to the description. So you can do this activity by pausing the video or either copy down this thing into the writing book, and either way you can find out what will be the structure. That's the only competency level that you have to pass in this lesson. So hope you got many ideas about this thing, about the cell and labeling part, how to microscopically find out what is the cell type and what is the shape of the cell, the microscopic slide formation. So hope you got many ideas. In the next session we are going to meet with the rest of this lesson, that will be the cell growth and cell division part.